Good day, everyone. My name is Annalisa A. Bukbuk, B.Ed. 1, Group 9. We are here to report the topic Gene Therapy versus Stem Cell Therapy. Our learning objective, our students should know the different and similarities of the gene therapy and stem cell therapy. Gene therapy consists of the introduction of genetic material into cell for the therapeutic purpose. Cell stem cells can be defined opportunity as cells that can be accurately self-renew and have the multifunctional generate of intermediate and mature cells. Genetic has to help come from the variety out sources and preventing those rocks can be much more completed than in supply assessing disease and frequency pattern. So majority of the health conditions are not related to a single genetic. Used to put result from the combination of inherited and environmental frequency or topic will highlight RAS factors and provide RAS assessment tools which can provide to healthcare decisions making. Because chromosomes and genes provide the introduction for your body's any change to those introductions have the potential to choose genetic diseases and negatively impact the health with diseases, the type of genetic diseases, how those diseases are inherited, and the potential health outcomes.
Understanding gene therapy. The expression of genetic condition is not always as simple as the amount of DNA present or the inheritance patterns of a single gene disorder. There are many factors that determine how one's genotype or genetic makeup correlates to one's phenotype or physical manifestation. Among these are penetrance, expressivity, mosaicism, and X inactivation. So, first is penetrance. Penetrance is a measure of how often a disease genotype correlates to the disease phenotype. For example, in a completely penetrant disease, every individual who is genetically positive for the disease would have clinical manifestation of said disease. In contrast, for diseases with incomplete penetrance, some individuals who are genetically positive for that disease may be entirely asymptomatic. Organisms inherit genetic information in a variety of ways that result in continuity of structure and function between parents and offspring. Suppose you as a child have a genetic disease, one that could be cured by diet or similarity straightforward therapy. Wouldn't the parents want disease diagnosed? Or take another example, perhaps two people are getting married. Wouldn't it be beneficial to know what chance their offspring have inheriting one or several harmful genetic traits? We are now armed with the different advanced procedures like PCR Southern blotting, RFLP mapping, and DNA sequencing. Different laboratories have already developed the ability to provide a very detailed look at our genetic materials. Is there a necessity to apply all these techniques to all of us? What harm might it do to have a complete picture of our genes? In totality, these procedures do not insert new genetic materials into cells. No changes of any kind are made. We are only provided with information about what is already there.
Cases of prenatal or perinatal diagnosis of a birth defect or an enzyme deficiency is the one situation where DNA diagnosis could be of immediate beneficial use. What most emphasized is that the information to be gathered by the new technology should be private and used by the individual who DNA has been read. A person DNA sequence to my mind should be protected by law from distribution to another parties without the individual specific permission. This goes the same with physicians who have obtained such information, who should also treat it as the any privileged material. The introduction of foreign genes into organism doesn't seem to be dangerous. The resulting organism don't appear to be better able to reproduce than any another attempts to remedy human disease by somatic genes therapy. Also don't appear to pose ethical dilemmas, but the introduction of genes into the human germs line does the diagnostic procedure that genetic engineering alone are the very powerful. Before anything else, it will be reliability easy to correlate the presence of virus, DNA polymorphism with disease and another hereditary characteristic.